So good afternoon, everyone, and welcome to our Seminars in Ageing program. And it's really exciting. We're finally back in the room together. I know we have lots of people online, but, you know, technology is great now. We can do our hybrid sessions. So for those of us here, we have sandwiches outside that you can bring into the room and eat as we hear our very exciting speaker today. So before we kick off with our speaker, I'd like to acknowledge the traditional owners of the land on which each of us is meeting. And for us here in the room at NARI, that's the Wurundjeri Woi-wurrung people. So I would like, on behalf of NARI, to acknowledge the traditional owners of the lands and waters across Australia and pay our respects to elders past and present and to all Aboriginal and Torres Strait Islander people. So a little housekeeping regarding our online audience. At the conclusion of the talk, we're going to have some Q&As, and you can submit them throughout the seminar using the Q&A function. So you should be able to see that using the Q&A tab at the top of your screen, which should resemble two speech bubbles, one with a question mark in it. The chat function for this is turned off, so please use the Q&A to submit your questions. Obviously in person, if you'd like to ask a question, you just have to raise your hand and we'll just repeat it up the front here so that the online audience can hear. And of course, we record our seminars and that will be uploaded to our website within a few days. So for those people who want to watch later or have to leave early. But today we're going to be hearing from Dr. David Barry, which is very exciting for all of us here. Dave's a new member of our NARI team. He's been here a couple of months. He's a research fellow in the Clinical Gerontology Division, currently working on the IDC Improve project. He has a background in human nutrition and biomedical science and completed his PhD at Swinburne University of Technology on the relationships between muscle health, physical performance and gut microbiome in older adults. He has experience facilitating investigator-led and commercially funded clinical trials across a range of disciplines, including rheumatology, dermatology and gastroenterology, as well as healthy volunteer and vaccine studies. And before moving to Australia in 2012, and you'll notice when he talks, he has a slight accent. He was in clinical practice in Seattle, USA, with a focus on integrative and lifestyle medicine. So his research interests include nutrition, the gut microbiome, and physical performance in ageing. And today, of course, he's presenting on the gut microbiome and outcomes from his PhD research. So welcome, Dave.
Thank you. Let's hit go. Welcome, thank you for that. Yes, I do have a funny accent. Please don't hold it against me. I'm American, but I'm very thrilled to be here presenting with this opportunity to present on my research for my PhD at Swinburne University. And I hope I've got my placement in the camera right for all of our attendees at home there. So hello, everyone in remote attendance. Today, we'll talk about the clinical trial that I ran as a core part of my PhD at Swinburne University. We looked at the gut microbiome and associations between physical performance and older populations. The objectives were, this was essentially, it was a double-blind, placebo-controlled randomized trial looking at a targeted symbiotic in performance measures that were associated with sarcopenia. We aim to investigate the effects of this on human gut microbiota, its microbiome, of course, and these clinical aspects of sarcopenia. Behind the scenes, we were interested in these clinical measurements of sarcopenia, but it was really just looking at commonly evaluated physical performance measurements. I'd like to start by acknowledging the broad team that helped contribute to the success of this project. It was essentially sponsored and driven by Swinburne University. My core team was at Swinburne. All the clinical study visits were there. In fact, I just want to put a shout out in the room here that we've got one participant from the clinical trial that's actually in attendance here today. That's just a very welcome surprise. I was not expecting that, but welcome and wonderful to see you again. But all the study visits were run there at Swinburne University, the Hawthorne campus. And we partnered up with a wonderful team at a number of different domestic and international institutions here, namely Deakin University. We had a lot of wonderful colleagues in the Institute for Physical Activity and Nutrition at Deakin. We leaned heavily on their expertise and also their DEXA scanner because we didn't have one at Swinburne. We wanted to get body composition as part of the outcomes for this trial. So we liaised closely with some investigators over at Deakin to conduct pre and post DEXA scans for this study. RMIT, we had a good group of some physiotherapists at RMIT up in the Bandura campus that helped us design the methods around our muscle ultrasound part of the protocol that we'll hopefully have time to talk about a bit today. La Trobe University, we had some great researchers both in the applied and environmental microbiology department, as well as the metabolomics and proteomics platform that helped us with our analyses for the serum and stool metabolomics and the microbiota analysis. Hugely involved, and we'll talk a little bit more about that later today because some of those results are still pending. But can't go through this presentation without giving great acknowledgement to our industry partner for this particular project, Lallement Health Solutions. They're out of Quebec, Canada. Wonderful company to work with. We've been absolutely pleased with our collaboration throughout this whole process with this project. Lallement provided in kind, of course, the study supplement, the investigational product or the symbiotic that we'll talk heavily about today, as well as provided some funding to help obtain the DEXA evaluations, the body composition for this particular trial. Otherwise, we wouldn't have been able to have the resources to do that. It's a big study, and Lallement was centrally involved right from the start. The design, the methodology, yes, but certainly the strain selections that we'll use for the study supplement or the investigational product in this trial. Just going to throw a disclaimer out there right from the start. Lallement, wonderful company to work with here. They are considering holding patent protection rights for the IP, literally the investigational product or the intellectual property for this study. So I can't name the actual strains used in this multi-strain symbiotic. So you'll notice a very big omission there when we get to those slides later on. Apologies in advance, but this is in honor of our agreement with Lallement Health Solutions. Great company to work with, excuse me. Cool. Jump right into it. Sarcopenia, many of us might be aware. This is generally the loss of skeletal muscle, mass, and function. Used to be considered most commonly with advancing age that has impacts on physical performance. So as we mentioned, sarcopenia can be diagnosed with impairments to physical performance, but following objective measured evaluations in low muscle strength and low muscle mass. So we'll lean towards the EWG SOP criteria for sarcopenia. There are a number of different ways of defining sarcopenia around the world. The Asian Working Group, the FNIH in the United States, but the European Working Group in sarcopenia in older persons, the revision 2 from 2019 is what we'll default to for this particular protocol, excuse me. Their criteria, unless I'm mistaken, it's been updated since 2019, but probable sarcopenia is identified by criterion 1, which is diminished strength. Yes, most commonly obtained via hand grip strength or repeat chair stands. Diagnosis of sarcopenia is confirmed by objective measures of decreased muscle mass, most commonly obtained with DEXA, MRI, CT, sometimes BIA, depending on the scrutiny of the journal you're trying to publish with. However, sarcopenia is considered severe. This is severe sarcopenia if all 3 of these criteria are present. So decreased muscle strength, grip strength, chair stands, some objective measure of muscle mass or quantity and quality, also in place with impaired physical performance, most commonly from components of the short physical performance battery, timed up and go. There's a number of different evaluation methods for looking at or teasing out physical performance deficits. So if all 3 are present, that says, yes, sarcopenia is most certainly there, it's present, can be diagnosed, and it's also considered severe. Does that make sense? Muscle mass, we're all aware that it can decrease depending on what publication you're referring to here, but there's wide estimates that muscle mass can decrease at the rate up to 2% per year after the age of 50. Disclaimer, obviously, we all know it really depends on what population we're speaking about, what ethnicity, background, the cohort. They're institutionalized, community dwelling. There's a lot of variation in the literature there, of course, but one thing that we can agree on, if we're lucky to live long enough, we can expect to see some deterioration in muscle mass and muscle strength, right? We all acknowledge that. We all know it happens. And some of us, it happens more progressively, a little bit earlier than expected. Some of us are nice and vital and strong and healthy until the later years in life. You've got good longevity of health span. This concept of sarcopenia is still not hugely widespread. It's still kind of being determined and commonplace in diagnosis and clinical treatment paradigms, partly because we don't have a really good handle on how we define it, right? But we all kind of assume, yes, if we're lucky to live old enough, we'll have some deficits in strength, mass, perhaps, as well as physical impairment. There we go. So sarcopenia has been associated with an increased risk of falls, disability, loss of independence, hospitalization, as well as increased mortality. Depending on the journal source, sarcopenic individuals have a significantly increased risk of mobility limitation, hospitalization, and even death. Sarcopenia is linked to several chronic diseases and associated with a higher chance of falls, fractures, as well as hospitalization. As I mentioned before, we collectively are still defining. We don't have a good handle on what this sarcopenia thing is, how it behaves, how we even diagnose it. A lot of evidence over the last 15 or 20 years from a number of different research groups have elucidated a lot of different potential mechanisms that might lead to this impairment in muscle mass, strength, and physical performance, the sarcopenia, right? Neuromuscular or mitochondrial dysfunction, yes. Insulin dysregulation, certainly. Chronic inflammation, that inflammation with aging, so that chronic inflammation. Lifestyle factors, certainly. Protein imbalance, so dysregulation between muscle protein, anabolism and catabolism, and the muscle protein synthesis states here that is, for whatever reason, perhaps impaired in certain individuals at certain stages of life. Welcome. Hello. Hormonal changes, certainly. A lot of research has looked into testosterone as well as impaired growth hormone sweeps. That circadian rhythm of the pulsatile nature of growth hormone, cleaning up the abuse of the day, that is certainly impaired as we age. That's one reason why it takes less to knock us over and a little bit longer to get up as we're getting older. But one thing that we'd like to focus on is the nutrition that I kind of skipped past, as well as the lifestyle factor. So much evidence from different research groups around the world looking at, well, what can contribute to prevent or certainly treat this sarcopenia. Yes, it's highly prevalent, certainly. Wide variations have been reported in the literature. Again, it just depends on what journal, what population, what group, what nation even. Could list a number of different studies here, but prevalence is hard to get a handle on, again, just because it really depends on the diagnostic criteria being used, the populations that those may or may not apply to so closely, whether they're in hospital, community dwelling, you name it. But it's clear that sarcopenia incidence is quite high. Prevalence can be as high as 50% in some studies, depending on what cohort they're looking at. General population estimates, again, wide variations there. 4 to 10% might be considered kind of average. A lot of costs. Oops, did I skip a slide? No. A lot of costs associated with this sarcopenia thing: impairment, physical performance deficits, falls risk, fractures, immobility, hospitalizations because of some of these scenarios. Negative outcomes resulting from declines in muscle mass and function can contribute to these high percentage of healthcare costs. In the United States, this is an old study. I do apologize for that, but Jansen's work back in, I think it was 2004, estimated annual healthcare costs that can be attributed to sarcopenia somewhere upwards of $18.5 billion. That's just massive amounts of zeros there, which can be correlated to about 1.5% of the total healthcare expenditures in the United States back in that year, 2004. It's shocking. Let's talk about human gut microbiome. I'm trying to push through this so we can talk about the data from the study here. So my apologies for kind of speeding through a bit. Some of this might be common knowledge for people here, but yes, this wonderful microbiome that we all have. A number of different types of microbiomes exist in and on the body: the skin, the oropharynx, all of the pulmonary system. Certainly the GI tract is the largest and probably most studied, best established. But the human gut microbiome is composed of as many as 100 trillion microbes. This vastly outnumbers the number of cells that make up our body, of course. The corresponding genome of this microbial community is termed the microbiome. So there's a lot of, a lot of people interchange the terms microbiota and microbiome. It's just kind of a pet peeve of mine, but they are kind of distinct differences, right? Collectively, the gut microbiome encodes about 150 more unique protein encoding genes than the human microbiome does, which is fascinating. Last count, somewhere, what, 20,000 genes in the human genome. These microbes that are occupying us, that we're in symbiosis with here, vastly outnumber the genetic potential than we ourselves have. It's just mind-blowing when you stop to think about it, right? Where was I going with that? More than 1,000 distinct bacterial phylotypes have been identified. Give or take, probably 90% can be attributable to two major phyla, so Bacteroidetes as well as Firmicutes. This gut muscle axis has been kind of a concept for the last 15 or so years, spearheaded by a number of researchers, mostly from throughout Europe, these wonderful pioneers in this research looking at the associations between the gut microbiome and muscle, right? Clearly, it's not a direct link, but there's a lot of evidence to support there's this connection, much like the gut-brain axis, which is just much further along with research. We've had much larger number of researchers around the world looking into this gut-brain axis for much longer. We're just that much further ahead. But the gut muscle axis, or the GMA, is kind of catching up, right? And it's really quite fascinating. That's where the rest of this talk will be kind of centered around. But evidence does support a bidirectional relationship between skeletal muscle as well as the gut microbiome. The cause of sarcopenia is likely multifactorial that we mentioned before, but recent evidence suggests that alterations in the gut microbiome may play a central role. That's this concept of the gut muscle axis. That's what we're also trying to therapeutically target with this clinical trial. Multiple cross-sectional studies centered on older subjects have found associations between the fecal microbiota composition as well as clinical aspects of sarcopenia. No surprise. Just one of a number of different studies that looked at this over the last 10 years or so. The ELDERMET study looked at 371 cohort subjects, their stool analyses, and found a very strong association between the presence of frailty in community-dwelling older individuals and comparable microbiome profiles of aged care residents. That was a really fascinating study. It was back in 2016. Okay. Beyond the scope of today's talk, but dysbiosis, we've all heard of it. We don't really have a good definition for it, but intestinal dysregulation, right? So what's going on with this profile, what we consider normal versus abnormal? What bugs are meant to be there? What microorganisms are present or absent, and what does that profile look like with each of us individually? And how might that be imparting clinically manifested disease, right, or illness? Certain things we know every day, you can't watch the morning news without some mention of microbiome, its associations with some health, skin health or anxiety or whatever. Yes, it's very common. But we know a lot of different environmental factors can contribute to the profile of bacterial organisms that are present in the gut, this microbial profile, which can be either positive or negative, and impart good or bad changes to the microbiome profile. Oops, wrong way. So this concept of dysbiosis, there we go. If present in an individual, again, that's open to differences of opinion of how we define that, but it can certainly lead to some of the things that we mentioned before that might be causes of sarcopenia, right? Chronic inflammation, persistent chronic systemic inflammation, endogenous biomolecule disruption. We got perhaps greater susceptibility to infection, adverse drug reactions, which I find really fascinating, altered profile of metabolism, certain pharmaceutical loads that are on board in certain patients for certain conditions can be altered by dysbiotic profiles, which I find just really fascinating. But malnutrition is also a result of dysbiosis from impaired nutrient absorption, biomolecule disruption. Okay. Emerging evidence, predominantly in preclinical models, suggests that disruption of the balance between microbial communities, such as the depletion of symbionts, as well as the overrepresentation of bacteria that we'd rather not see present, these pathobionts, this can lead to this concept of dysbiosis. And there's certainly a lot of evidence that supports those correlations. Yep. So as we age, we can expect to see shifts in the microbial profile. So the relative abundance of certain bacterial species, we know that shifts across the world. And this is just kind of going to happen. Yes, it's sort of a profile of aging. But relative abundance can go down. The microbial diversity, so the representative samples, how many different types of species out of those 1,000 we'd expect to see are present and to what extent. So that's relative abundance and composition, yeah. The protective taxa that's represented by those bacteria might very well get shifted or go down as well. With that, we might see the weeds growing a bit more, right? Those increased relative abundance of the pathobionts, the ones we'd rather not see. They usually don't cause problems unless they are in significant amounts. They can contribute to poor outcomes. I really like this slide from Gosh 2022, looking at kind of that concept of longevity and healthy aging. So with chronological age across, we can see the pathobionts might very well increase. Protective taxa can decrease, and that can lead to increases in inflammation or frailty and degeneration. Not so much sarcopenia per se, but it was really quite applicable. I found it fascinating. Gut microbiome is a modulator of healthy aging. Kind of already talked about this, so I'll push through it. But changes in the gut microbiome with aging can lead to shifts in the microbial-derived metabolites here. So these are those short-chain fatty acids, right? Butyrate, propionate, acetate, in the respective acid forms. Very, very central. We believe that's many times the underlying mechanism for some of these changes that we can see. Just disrupted or impaired short-chain fatty acid production. I was going to mention something else about that. Certainly, it's not just short-chain fatty acids, but microbial derived metabolites. These can include, tryptophan derivatives. I mean, there's a number of all the nutrients and vitamins and minerals that are created, synthesized by the hopefully present microbial populations within our gut there. Yes, serotonin, right? Largest pool of serotonin is present right there in the GI system. But anyway, increased intestinal permeability at the local level, as well as this chronic systemic inflammation. Older adults are the fastest growing global subpopulation, so we can expect to see these impaired microbial profiles increase collectively around the world, which may very well lead to increases in expected rates, incident rates of sarcopenia as well, unfortunately. Good news is we think we can do something about it, As a summary, before we pitch into the actual clinical trial notes, sarcopenia is highly prevalent around the world. Yes, we don't have a good handle on exactly what those incidence or prevalence rates looks like, but we know it's there. We know it happens. We know it occurs. We're getting a better handle on how to describe it, define it. It's expected to rise because that global subpopulation is going to increase. The population's increasing. We're living longer, so we expect to see those incident rates increase as well. There are currently no pharmacological treatments for sarcopenia per se. There's a number of, maybe a dozen or more different medications, pharmaceuticals that are in phase 2 and beyond trials, I believe, around the world, but none of them come to market. It's kind of interesting. Aging is associated with changes in the microbiome, yes, and there is evidence to suggest that microbiome therapeutics could play a role. That's kind of the reason we brought this project together. Okay, let's talk about the study. Sorry, how are we doing? 20 minutes already. I'll try to talk a little bit faster because this is the fun stuff. So this particular clinical trial, this was a randomized controlled, placebo-controlled double-blinded study. Yes, we wanted to evaluate the effects of a multi-strain symbiotic on functional parameters associated with community-dwelling individuals. We aimed to enroll 74 eligible participants into 2 groups, the intervention of control group, in this case, the maltodextrin placebo. We were going to give them this oral symbiotic or placebo every day, once a day for 16 weeks. So it's a 4-month intervention. We stratified them 1-to-1, yes, if they were eligible, and we enrolled them into the study. They were block randomized on gait speed and hand grip or muscle strength. We'll talk about that later. Back to my disclaimer at the beginning. Our wonderful partners, Lalaman Health Solutions, are considering pushing forward with this actual product partly because of the success of this clinical trial, which of course needs to be verified, confirmed, and followed up with. But it was just kind of surprising, to be honest, the results that we got. We'll talk about that in a moment. But we can't name the specific strains. There were 3 of them. They were reasonably commonplace. We expect to see them. And they were in different profiles, different ranges. We had, 50%, 40%, 10% of 3 of these different bacterial species, okay? What I can say is we included inulin in the treatment, in the investigational product at a dose of 900 milligrams per day. This strain was in an oral sachet, a powder form. The placebo was maltodextrin, as we mentioned before. And the actual products themselves were cultured over it was about a 6 or 9 month process. It took a long time, but they were actually developed in France in the Roselle Institute, which is a branch, the microbiome formulation branch of Element Health Solutions. So getting that temperature sensitive live strain organism through TGA into Australia for this clinical trial was a bit of a logistical nightmare, but we got there, and it was a really fantastic project. The IP and matching placebo were in these little white sachets. They didn't taste all that bad, did they? I'll ask the participant in the room if you're still awake. But we asked them to take these sachets, keep them in the fridge, pull them out, rip them open, dissolve them in a bit of water, take them as close to possible at the same time of the day, just to try to replicate that 24-hour dosing cycle. This was placebo or the intervention, again, once daily for 16 weeks. So it's a 4-month intervention. Yeah, that's what we talked about. So here's the study profile, 5 study visits over 22 weeks, give or take. The screening visit, which is approximately 14 days or less, we tried to keep it right at 7 days. Then we had the intervention over 16 weeks, so it's a 4-month intervention, certainly baseline, mid-intervention at 8 weeks. Then we had the end of treatment visit at 16 weeks. We had a follow-up visit at week 20, so 4 weeks after that participant finished their 16-week intervention. The screening visit, obviously, first thing we explain the study, we obtain informed consent. We lost quite a number of participants through screening failures at this particular visit, but we had a very robust eligibility criteria for the study, and we'll explain that in a moment. But we obtained informed consent. We ran the participants through a familiarization session with the performance measures, so all these things that we'll talk about in a moment, but just making sure they are able to comfortably do these things and do them correctly the way that we want them to do. And we also organized the first DEXA scan, the pre-treatment DEXA scan. If we believe they'd be eligible, we bring them back a week later for that baseline visit at time 0. Oh yeah, right. We sent them home because they were not consented yet for the screening visit, so we couldn't ask them to fast. We couldn't collect blood at that visit. So we did all that at baseline visit a week later. So we informed them of all the rest of the study procedures for the rest of the trial. So every visit you'll come in fasting. Please wear shorts because we'll do some cool testing and some muscle ultrasound. But here's your little stool kit. Please collect a stool sample. And we gave them very clear directions with how we wanted them to obtain these samples, protect those samples, bring them into us frozen and all that. So there's a lot of instruction. And we had a really wonderful cohort of participants. They were very engaged. They were really willing. They put up with everything we asked them to do. It was truly phenomenal. It was amazing. But yeah, and we gave them the 3-day diet diaries and we explained how we wanted them to capture that information for the 3 days leading up to each subsequent visit. Methodology. So the study population, I think I mentioned this before, but we aimed to bring in 74 participants, so 37 per arm, treatment control. These were 60 to 85-year-old male and female community dwelling individuals. So out there in the community, right? So these were specifically not aged care home. They were not sarcopenic participants in general. These were just folks from the community, very engaged. We had a lot of eligibility criteria, as I referred to earlier, but some of the core ones here, you had to have a BMI between 18 and 30 with a minimum body weight of 40 kilograms. You had to be able to do the physical performance testing. That was quite clear. So anybody with musculoskeletal disorders or certain arthritis, they were just kind of unfortunately ruled out of the study. And of course, lower upper extremity fracture in the past 6 months, hip replacement surgery in the past 12 months. We just want to make it very clear that we can compare data. One other big one that we actually dropped a couple people from, the use of pre and probiotics for 4 weeks. We allowed a 4-week washout. So if they were coming into the screening visit and they were like, yeah, kefir and probiotics and whatever, say, that's wonderful. Are you willing to stop for the duration of the study? If they were, sweet. We walked them through a 4-week washout, brought them back in for rescreening and went forward from there. Antibiotics during the course of the treatment, the whole clinical trial there was one of our exclusion criteria. We actually did lose one participant who unfortunately had I forget what infection that required antibiotics. We had to discontinue him. So I'll speak about the methodology, but we were able to publish. The central project team from Swinburne put a protocol paper together outlining the methods that we put together for this particular trial, and we were registered on ANZ CTR. So this study looked at 4 broad goals, and I'll refer to for the rest of the presentation today, the primary and secondary outcomes for the study. Primary are the physical performance outcomes we were most centrally interested in, right? the effect of this multi-strain probiotic versus placebo on physical performance measures in older individuals, community dwelling. Secondary aims, we're looking at the effect of that strain symbiotic on muscle mass, as well as microbiota composition and the associated metabolites. Exploratory aims, and I don't think we'll have time to talk about them today, but I would love to be able to. We looked at the use of muscle ultrasound, the utility of ultrasound compared to DEXA and BIA for body composition analysis, specifically with muscle architecture, right? Muscle quantity and quality, which is just really fascinating. But I don't know if we'll have enough time to talk about today. As a clinical trial, we ran some safety panels there as well. We wanted to assess the effect, the tolerability, as well as the safety of this IP on our participants. To achieve these aims, use our outcome measures for the primary, very straightforward, hand grip strength, 4-meter gait speed, standing balance, repeat chair stance. So those are components of the short physical performance battery, as well as timed up and go. The secondary aims here, looking at the muscle mass, as well as the microbiota profile and the changes across the intervention period. So body composition, yes, we looked at fecal microbiota composition and diversity, so alpha and beta diversity matrices, as well as the untargeted fecal and plasma metabolomics, some of which are still pending, unfortunately. Lallerman is helping us with the qPCR for probiotic strain recovery, just to prove like those strains were present, they were increased over the duration of the study, and we can clearly map that. Exploratory aims. We ran a whole bunch of different questionnaires throughout the study at different time points, but two of the central ones with respect to tolerability were the GSRS, the gastrointestinal symptom rating scale, as well as the constipation assessment scale. So our participants got really used to filling out these questionnaires for us, as well as, of course, the 3-day diet diaries at each time point as well. But muscle architecture and morphology was assessed via ultrasound. I hope I had a chance to chat about that today. The safety profiles, we looked at basic changes on outside clinical variation in chem panel and CBC. Just for the 16 weeks, so the baseline intervention, mid-intervention in a 16-week visit. And of course, we tracked any adverse events and of course serious adverse events till the end of the treatment or the end of study completion, respectively. Fortunately, it was shocking how clean the study was, how minimal we had a negative impacts. So hand grip strength by a dynamometer. The 3 components of the short physical performance battery: gait speed, standing balance, and the repeat chair stance, as well as timed up and go. These were the methods of assessment for our physical performance. Sorry, what?
I was going to say,
Speed up.
Yeah.
There we go. Yeah, I'm watching the clock. So we use BIA as well as DEXA at 2 time points. 2 time points for DEXA for looking at body composition, as well as BIA in office there at each of the study visits, and the exploratory outcomes for muscle morphology and architecture. So I'm a stickler for method. I do need to take a moment to explain this here because it's really quite central to our study. Those of us who do this type of research or are interested in it, or clinicians in the room or in the attendance remotely, we are aware that there's a wide variation of the way that people could do certain assessment tasks, right? Hand grip strength, we all do it a little bit differently. And that, for research purposes, drives us a bit crazy because it limits our comparability of our data, right? So with respect to hand grip strength, for our study, it was all laid out in our protocol paper, and we try to follow the consensus out there. But our study, we used a JMAR analog dynamometer, yes, set to position 2. For everybody, unless they had really big or really small hands, we made that adjustment. But I think it was across the board, position 2. They were seated. They were at 90 degrees of elbow flexion with the thumb up. We asked them to squeeze maximally, yes, for 6 seconds, 3 times with 1 minute rest between. We used their dominant hand. We did 3 tests with that 1 minute break between. We analyzed both their peak performance as well as the average of 3 attempts. Okay, so short physical performance battery, the composite measured 3 tests. Gait speed was over 4 meters in our case. Again, there can be wide variation for how this is performed. We all know this, but just to be clear, we started our time element at the 3-meter mark and finished that at 7-meter mark. So over a 10-meter course, we had our participants begin at the starting line and said, go ahead and walk at your habitual normal gait speed. They didn't know what we were doing. We were standing on the side there. We started the timer when they crossed at the 3-meter mark, finishing at 7 meters. So we captured habitual walking speed over 4 meters. Most studies, not sure why, but they'll start at 0.0, say, please walk across this 4-meter line. They started at the beginning when they go, and that includes, of course, the initiation or the acceleration phase that arbitrarily inflates their walking speed or their time over 4 meters. So that's not good data. But anyway, stickler for the rule, our protocol, not a rule. Repeat chair stands fairly straightforward, but these were, yes, hands crossed, seat. We use the same standard seat for every participant here too as well. Nice and padded, but it was very comfortable and standard across the process. And we asked them to sit fully and rise fully. So hips and knees fully extended. We just track that. And that's part of why we ran them through all these physical performance assessments in that screening visit to very clearly explain what the expectations are, so they followed it consistently at each time point. SiA and DEXA, I think we know what they are. Of course, they were fasting for each. And we did them at the beginning of the visit. Exploratory outcome measures. This was the muscle architecture. I won't have time to talk about this today, but perhaps in a future session. But just briefly, we did run muscle architecture on their dominant leg. We used to analyze for muscle quantity as well as quality, because these are emerging areas of interest for folks looking to assess muscle architecture. It's portable, there's no radiation, it's pretty cheap. A lot of people can do it, it's really simple, it's painless. Like there's a lot of advantages to using ultrasound. And we use it all the time, diagnostic and therapeutic ultrasound, yes. But specifically for assessing muscle architecture, with looking at quantity for detection of sarcopenia or impairment, right? That's not clear yet. And certainly not what's used. It's not standard. It's not the base. It's not DEXA. It's not CT. It's not MRI. But it's also nowhere near as expensive or logistically prohibitive. Not everybody's got a DEXA scanner. It's expensive to send them for an MRI to get muscle body composition and look at the muscle mass, right? But ultrasound, anyway, I'm passionate about it. It's an area of interest. And unfortunately, we probably can't chat about it. That's a bummer. Okay, biological samples, yes. Starting at baseline visit for the rest of the study, we obtained blood samples here. Some that we sent out for safety labs, yes, but we were able to biobank a lot of serum and plasma for our serum short-chain fatty acid analysis for GCMS, as well as we anticipate doing some of our inflammatory pathways, which we didn't have funding for at the time of the study launch, and also some markers of inflammation, indirect markers of intestinal permeability, LPS and zonulin, and so forth. We collected stool at each visit, again, same thing, but we were running the qPCR strain recovery, as well as stool metabolomics and the microbiota profiles. Some of those are still being run by a proteomics and metabolomics lab up at La Trobe, as well as the applied microbiology groups there. Wonderful teams. Okay. Because we all know that physical activity and diet can significantly impact the gut microbiome, which has its attendant results on skeletal muscle performance and physical performance, we tried as best as possible to monitor for changes. We asked all of our participants coming into the study, please, so far as possible, make no changes to your current diet and your current activity level. We don't care what they are. If they're training for a marathon right now, that's fine. Hope you can keep up with that, but please make no changes throughout the 16 weeks that you're on the study. Same with diet, as best you can. And we tried as best we can to monitor for that. So 3-day diet diaries, 3 days preceding each follow-up visit. We did the DQES at baseline only, just to get a nice deep dive of their intake patterns for nutritional analysis. For physical activity, we couldn't afford accelerometry or other devices to really track movement patterns and things like that, which would be a really cool thing for future follow-up studies. But we did the physical activity scale for the elderly at each time point. Quite simple, quite easy. It does give us something to run off of. So here's the outline. We collected everything at every visit. DEXA scan at 7 days pre or post from baseline, and then of course the second DEXA scan was at 16 weeks. IP accountability, because that's really quite important, is your sachets. We asked our participants to take one once daily each time, same time of day, and bring those sachets back at week 8, week 16, so we can do our IP accountability checks. We asked them to bring back their empty and unused packages, and we just did that accountability there. So good news is we had incredible compliance with our IP. Okay. Recruitment. We brought 118 participants into the study. We lost about 48 with screen failures or just chose not to participate. We were able to enroll 70 participants on the study. Ended up being 34 in the placebo group, 36 in the treatment group. At the end of the study, at the end of 16 weeks, by that follow-up visit, we had lost 6 total participants by design, but it was just an even 32 in each treatment group. It was really, really wonderful for data comparison purposes. Again, not by design, it's a bit bizarre. Baseline characteristics, we had no significant differences between groups at baseline. There was one that was trending there for SPBB score, interesting, slightly higher in the placebo group. So let's talk about outcomes. These are the physical performance outcomes. Hand grip strength, first and foremost, just looking at monitoring hand grip strength. The treatment group, there was a trend for increased hand grip strength. On average, of course, mean in SD was 1.2 kilograms higher at 16 weeks in the symbiotic group. And this was significant compared to placebo over time. With respect to the short physical performance battery, this is the overall score. Yes. So there was a trend increase for the symbiotic score from baseline over placebo. That was significant, as well as we had both the time as well as the interaction effects were both significant over 16 weeks. This is the composite score for short physical performance battery. So of the breakdown of the components, because we know that that's the SPBB score is contributed to by gait speed, chair stands, as well as the balance score, which we didn't talk too much about. But there were some significant scores across some of these parameters, specifically the repeat chair stand score, as well as the repeat chair stand time. Again, improvements in the symbiotic group versus placebo, as well as the gait speed, the 4-meter gait speed, the way that we assessed it. There were improvements that were strongly significant in the symbiotic group over placebo. No significant changes in overall gait speed score, which isn't all that important, or the balance score, which is certainly important, but it wasn't significant between the two groups in our study. Timed up and go. We kind of skipped past this, but very simple test, having a participant stand up, walk at their habitual gait speed around a cone 3 meters away, and come back and sit down. So start to stop, finish, looking at that time it takes them to do that. There are very clear normal values for men and women. So we did that at every visit as well for all of our participants, and there was a significant change both within group as well as between groups for improvement in the timed up and go score with respect to the sarcopenia. So I didn't show it on any of these slides here, but many of these results were carried over at 20 weeks. Remember, we had 16 weeks of intervention. We captured each of these assessment measures at each of the study visits, but then we had a 4-week follow-up just to see, did it persist? Did it continue to increase? Did it immediately fall out? Did it plateau? Like, what happened? With logistical limitations in time, this was part of my PhD, so we couldn't just keep going on forever. We would have loved to have seen longer follow-ups, certainly, not just 4 weeks out. And certainly larger numbers and multi-center and all that. But interesting of note, most of these changes, I didn't show it on these slides, they were carried over those 4 weeks. So because it's so important, here's the snapshot of pre and post changes in just some of the core elements of habitual dietary intake that we track for our participants. And at the bottom here, the physical activity score, the PASE score. So there were a few things that were significant. You can see in the symbiotic group, most of these were trending down if you look closely at the data there. For example, fiber intake, which is interesting. Fiber is fuel for those gut microbes, right? They love fiber. That's what they do. They metabolize that and create these wonderful short-chain fatty acids. So we're like, huh, that's weird. The group that had improvements that we believe were driven by some of these changes, these metabolites, actually had less fiber over time. So that wasn't it. It wasn't a change in diet that might have contributed to that, as best we can determine. But also protein, so grams per kilogram body weight, it did trend slightly down. It was significant within group. But across group, the far column there on the right, that was the interaction effects here. Again, no real changes. Like it was good. It was surprisingly good that they were well-behaved participants over the study. Yep. I think there was something else I wanted to say about that. Okay, body composition, no changes. I'll just save time right there. So this was BIA and DEXA. These data right here are for the DEXA results because they're a little bit more impactful. surprising, considering we had so many significant improvements in the symbiotic group with all the physical performance measurements that we were assessing over time. When it came down to looking at the core data for muscle mass and body composition changes, not a peep, no changes. Probably not surprising when you think about it. I mean, it is a symbiotic, it is only 16 weeks. You know, it's quite a soft therapy, if you can give it that way. So we were surprised to see changes regardless. We're happy with them, of course, but we looked at the body composition data over time, and there were no significant changes. Interesting. I wish we had time to chat about the muscle ultrasound, but I'm feeling it. That it's 12:46. Okay. Potential mechanisms, just briefly. Regulating energy metabolism, mitigating oxidative stress-induced damage, as well as modulating immune responses might very well lead to some of these perceived changes. There's a lot of different research groups out there that have provided evidence to support those thoughts. The short-chain fatty acids are what we believe are some of the mechanisms behind that, but these are still pending, as the table kind of suggests above there. Unfortunately, we're still waiting on some of the results for our alpha and beta diversities to see exactly what profile changes we saw with diversity and composition, as well as some of the stool short metabolomics. Watch this space, you know, but we believe we're expecting to see some changes in some of the short chain fatty acids in particular, butyrate being central among them. A lot of evidence supports that changes in bifidobacteria in particular can lead to increased butyrate, and that has pretty well a substantiated impact on muscle health, exercise recovery, muscle anabolism. But that's what we believe some of the mechanisms are. But we're waiting on some of our wonderful groups up at the proteomics and applied ecology labs at La Trobe. Future directions, those highlighted in red, are ones that we've got biobanks of, frozen tons of vials of frozen serum that we don't have current funding for. So if there's anyone in the room or anyone that's listening remotely that's interested and got a funding source, let's talk. Let's get together and put a team together in a project where we got some samples that need analyzing. We've got wonderful teams that are capable of doing it. And that could help us answer some of these outlying questions that might help us explain some of these observed benefits that we saw. Other directions that we'd like to go, larger confirmation study. Clearly, we saw some rather surprising results across the board looking at improvements. So this multi-strain symbiotic was able to show significant changes in hand grip strength, short physical performance score, subscores, timed up. It was across the board. Frankly, I was surprised. We did an interim analysis when we were getting really close to the end of enrollment. I was like, let's just have a look. It's all blinded data. Ripped it out, had a look at it, started running it through SPSS. I'm like, oh my God, one of these groups is showing some significant trend. Please let it be the treatment group. Please, you know, so we're testing our hypothesis. At the end of the study, we unblinded everything. We ran. I was like, oh my God, wow, it's across the board. Almost everything we looked at with respect to our primary outcomes were improved or bettered. Yeah. Surprising. Shouldn't have worked that way. Can't explain it. Clearly, we need to confirm that. We need to follow up. So further studies, international, multi-site, different demographics, different groups, that would be really helpful. So let's put a team together and look at that. I'd love to explore personalized microbiome therapeutics as well. So getting samples, we've got labs around the world that can take a look at that sample, say, what are you high in, what are you low in, what could benefit here with respect to your clinical conditions, scenarios, your age, gender, all that, and target specific microbiomes to influence those. Okay, I get the sense that's probably it. Thanks to the team. Huge thanks to our colleagues in Australia, Canada, France. They're listed there, of course, from RMIT, Deakin, and La Trobe universities here within Australia. Fantastic squad to work with, as well as our wonderful industry partners there in Canada and France, the folks from L'Allemand and Roselle Institute. I think that's a thank you.
So thanks, Dave, and certainly really interesting findings, and it definitely does bring up a lot of questions. I think you can ask questions there. Yeah. Is there any questions in the room to start off with? Yeah, thanks.
So you found significant differences between the groups on, let's say, timed up and low. But if I read the slides correctly, the actual magnitude of the difference in the time that window was something in the order of 0.2 seconds. Is that about right? Do you think?
Well, off the top of my head, probably. Yeah.
Intervention group and maybe, I don't know, slight reduction or increase in time for the control group.
Right.
So yeah, I'm just thinking about how a change of that magnitude, the size of that change compared to, say, an exercise intervention. Do you have any thoughts on that?
I do, certainly. There's a lot of evidence to support physical activity is powerful, certainly powerful. Sorry, I'm not sure where to stand here. I don't want to muscle you guys out. Certainly physical activity first and foremost, resistance training, combination therapy, high impact intensity exercise, certainly. Like there's so much well substantiated evidence saying that is definite key. That should be core for therapeutics going forward to try to minimize the impact of sarcopenia or muscle strength deficits and all that. Fully agreed. I think I'd like to pick up on your point there with it was rather nominal changes in timed up and go, for example. Hand grip strength was a bit stronger, but again, we had a good profile, mostly female, but good profile between the treatment groups in our study. One thing that I didn't report on was how this was powered, looking at meaningful clinical differences, or what does it mean? What is 1.2 kilograms of increased strength over this group? Is that significant? Does that mean anything with respect to grip strength? Or what is a clinically significant change in SBBB scores? Those discussions are, if I understand correctly, still happening, right? And it kind of depends on group, definition of sarcopenia, ethnicity, background. Like we've got a lot of different ways to answer that.
Because I think the change in, say, you talk about the goal, 2 seconds.
Yep.
Like functionally doesn't translate to much if you kind of extrapolate it. So I don't know what the gait speed was like, before and after. I can't remember, but yeah, very interested to explore that in more detail.
Yeah. What does it mean? We can show that it's significant with respect to our 70 people, but let's blow that out. Does it matter? Exactly. Good point. Let's look at it.
Yeah, I guess there is minimally clinically important differences that you can potentially look at. But I guess what's interesting about this is that there is an effect, that there's an effect and potentially that you might not expect in a direction that you might not expect, which is, with exercise, it's clear, the mechanism. This is interesting. Yeah. Any other questions from the room?
And it was specifically no changes to exercise, no diet. So to your point, clearly we know tweaking up your diet, getting protein above 0.8 grams per kilogram or whatever, resistance exercise in particular, those are significant, profound changes, almost immediate and can be sustained and quite easy to implement. But we wanted to say, look, as best we can, let's just look at this change. It's one change as best we can. So it'd be really interesting to layer that, have multi-arms, like resistance training program on top of probiotics or something like that over time.
3 arms.
Let's build it. Let's build it.
So there's a question online saying around the decline in fibre intake in the active intervention group. Was that something that was
That's correct.
Might be the question around that.
We kind of mentioned it during the slide, Alice. Thank you for your comment, your question. But yes, it's true. We were surprised also to see that there was a decrease in fiber intake. Again, these are averages over time. So this is gram per, I forget what our measurement was, but the amount of fiber intake that we recovered from the analysis of the 3-day diet diaries in the treatment group was actually lower. Because we looked specifically for that, like was any of this benefit from changes in their diet, even if small changes. But the treatment group over time had more fiber. We know fiber can increase, microbial profile as well as the metabolites that they produce. That could explain some of the benefit aside from our interventions. We wanted to look at that. But we were surprised, like, wait a sec, hold on. In this small cohort, one site, the treatment group had less, like noticeably, significantly less fiber intake over the 16 weeks. So we're like, that's noteworthy, but good pickup. Is that the only question from? There was.
Yeah,
We'll get
we
back to
did.
this.
We asked them to put it in the Q&A.
David.
Okay.
Unmuted David.
I don't know. We did ask them to put it in the Q&A if they could, just because it's a bit difficult.
Use telepathy, David.
Yeah. So anyway, that's
While David's typing in his response, we've got a question here in the front row from